So I just wanted to take a few minutes to talk about some of the key challenges that we face in drug development for CMT. These are obviously not unique to CMT, but I tried to sort of distill down some of the specific problems. Obviously, we're not talking about the entire problem of the drug funnel and getting drugs to market, but things that are specific to CMT. So in terms of the first thing, and this really, oops, I'm having a, you want to advance the slide? In terms of the first challenge, I think this is just something that actually has already been explicitly discussed in terms of delivery. The peripheral nerve is a privileged space. It's not easy to get things into the place where they need to be to treat problems with peripheral nerve disease. There are two different barriers to drug delivery. You have the perineurium that surrounds the nerve fascicle, which is where both the Schwann cells and the axons are and where you need to deliver a drug to. And you also have a blood-nerve barrier, and that is formed by non-fenestrated capillaries. So these are different than the normal capillaries that are quite leaky. And so drug delivery is a major problem that needs to be dealt with in terms of treating nerve disease and CMT in specific. Next slide. So I think that another thing that we need to think about is, as also has been alluded to already, there are a lot of different forms of CMT due to different genetic mutations, and we may not be able to address every single subdisease specifically. So we need to identify common pathogenic mechanisms and look for ways to address those. And I just identified four that I think are the most promising and overlap among different forms of CMT. The first is inflammation. We can advance the slide. The second is activation of the integrated stress response, or UPR. We're going to hear more about some of these. The third is disrupted lipid synthesis. That's a new, well, it's something that's been known for a long time, but there's been some very interesting work recently in CMT1A on that, and that's what the figure there is. And the third is to identify the drivers of axonal loss and of regeneration. These are all potential mechanisms that might cause different forms of CMT and allow for treatment that is mutation agnostic. The third challenge is the availability of animal models and the relevance of those models. We often don't know, especially in CMT1A, I feel like we don't necessarily always know what the best model is to use for a particular problem. And I think that's something that the field needs to get together and perhaps produce some kind of position paper on what are the advantages and disadvantages of each of the available models. Something else that I'll comment on that I think is not appreciated, I think there is genetic drift in a lot of the animal model colonies, and I don't think that every C3 mouse, I mean, obviously JAX does a good job of making sure that their mice are what they say they are. But I'm not sure that what people have in terms of like C22, for example, in the different labs that people have been using, or I should say breeding for the last 20 years or so. I'm not sure that they're always the same mouse between labs. Availability, we talked about relevance a little bit, and cost of models is an issue, especially for industry partners. I understand that industry has a more difficult time sometimes getting the right models to do the experiments they need to do. And lastly, in terms of rodent models, I think that there's a problem of scale in terms of really the size of the animals and the longevity. So something that maybe we'll hear about a little later is the question of whether intrathecal delivery of a substance is going to get access to the entire peripheral nerve. Well, in mice, that seems to be the case, but I think it's an open question about whether in larger mammals that's actually going to be the case. In terms of cell-based models, go back, please, for a second. In terms of cell-based models, I think that one of the key challenges is, while we do have many papers on how to produce myelinating Schwann cells from iPSCs, the methods are still pretty inefficient and require long periods of time to produce even a small amount of myelin in culture. This is obviously more of an issue for studying the demyelinating forms of CMT, but I think while there are much more successful models of neuronal or based on neuronal differentiated iPSCs, you really aren't getting a full picture about any neuropathy unless you also have a myelinating Schwann cell. So I think it's an issue also for axonal forms of CMT. Last slide, please. I just wanted to say a couple of things. I'm no expert on clinical trials, but I think it's worth pointing out that there are some inherent difficulties in conducting trials in CMT. These are not unique to CMT. These really are shared. And I should say these, CMT is a very broad set of diseases. So what I'm talking about really pertains to many forms of CMT, but not all forms. But many are slowly progressive. And therefore, it's sometimes difficult to find sensitive, reproducible, and clinically relevant outcome measures, certainly measures that provide all three of those attributes. The effect sizes are going to be very small for a disease that's very slowly progressive. And so you either need a long period of time or a lot of patients to see a positive outcome. So that means that we need probably to have biomarkers that are more sensitive, reproducible, and hopefully they can be shown to also be clinically relevant, because a biomarker is not inherently clinically relevant. That's something that needs to be proven. The other problem is that children are probably, children with, for example, CMT1A, to take as an example, they're probably the test subjects that are most likely to show a positive result. If you have a treatment that actually modifies the disease, there's a lot of evidence to suggest that younger patients are going to be more sensitive and are going to provide a larger effect size. But unfortunately, it's unlikely that, especially for a slowly progressive disease like the ones where the children are they're going to be the most sensitive subjects, it's unlikely that children would be involved in early clinical trials. So these are just some of the roadblocks, I think, that we face in terms of conducting clinical trials. And with that, actually, I'm going to stop. I don't have that much time, so I don't know if we even have time for questions, because we got a lot of stuff going on.